السلام عليكم ورحمة الله وبركاته. مرحبا بكم ثانية في المحاضرة الأولى من مادة الكيمياء الصيدلانية العضوية المرحلة الرابعة. This is lecture 1 part 1 of the anti-neoplastic agents. طبعا مثل ما قلت لكم بالintroduction انه احنا راح نبلش اولا drugs acting directly on nucleic acid. حيكون مواضيعنا هي حتكون intercalating agents, non-intercalating agents which inhibit the action of topoisomerase enzymes on DNA, alkylating and methylating agents, chain cutters, and antisense therapy. These are going to be the subject that we are going to be discussing in the first lecture or lecture 1. In this part 1, we are going to be discussing intercalating agents. Intercalating agents are chemical compounds that contain planar, aromatic or heteroaromatic ring system. مثل ما جاي تشوفون هنا هذا هو planar aromatic or heteroaromatic ring system. Alright, these agents can slip into the double helix of DNA and distort its structure. مثل ما جاي تشوفون هنا بالفيجر هذه agents. That's why they call them intercalating agents. They can intercalate, slip itself into the double helix of DNA. مثل ما جاي تشوفون هنا بالمثال مالتنا اللي هو بالفيجر. Okay, هذا هو ال DNA double helix. Okay. Alright, and this is our agent. Once bound, the drug can inhibit the enzymes involved in the replication and transcription processes. Okay, once bound, so how they are going to be bound? First, they are going to bound by van der Waals interactions between the aromatic or the heteroaromatic planar ring system and the nitrogenous bases of the DNA. As you know, we have the nucleotides, and each nucleotide has a nitrogenous base, and that nitrogenous base has an heteroaromatic ring. So there will be van der Waals interactions between the aromatic ring system and the nitrogenous bases. Also, most of these agents have either ionic group or ionized charged groups like NH3 positive in this example, but this is just example, and or some groups that are capable of forming hydrogen bond. Okay, so it will also form ionic interactions with the other ionized group of the DNA, or it will form hydrogen bonding with other groups that are capable of forming hydrogen bonding in the DNA, like the nitrogens or the oxygens. Alright? Examples on the intercalating agent is the dactinomycin, or it's used to be called actinomycin D. Now they call it dactinomycin. I don't see the difference. But anyway, it's a naturally occurring antibiotic. It's effective anti-cancer agent in children. Well, actually, it's one of the most effective anti-cancer agents in children. As you can see from the figure, it contains 2 cyclic pentapeptides this one and this one, and it also has a flat tricyclic heteroaromatic structure. This one here. Okay, this flat tricyclic heteroaromatic structure slide into the double helix via the minor groove. As you know, DNA, the double helix DNA contains major and minor grooves. This one slides into the minor groove. Okay, it forms van der Waals interactions between the heteroaromatic system, the tricyclic heteroaromatic system, and, as we said, the nitrogenous basis of the DNA, and it also forms hydrogen bonding interaction with the DNA through the pentapeptides. Those pentapeptides will form hydrogen bonding, while this one will form the van der Waals interaction. Okay, this will result in a very stable complex. Okay, and as we said, it distorts the DNA. And in our case, or in the dactinomycin case, it will prevent the unwinding of double helix. Okay, now someone will ask why the unwinding is important. Well, why winding is important in DNA replication or DNA transcription? Well, that is one of the reasons why I asked you in the introduction to read the introduction from the book. Okay, and I suggest, and I highly suggest, that you read the introduction if you want to understand what is going on. Another example on intercalating agents is the doxorubicin. It is also a natural product. It contains a tricyclic system, tetracyclic system. Okay, the dactinomycin has tricyclic system, 3 aromatic rings. Now this one has tetracyclic system, 3 aromatic rings, 1, 2, 3, and a 4th ring, but it is not aromatic. Okay, so it contains a tetracyclic system where 3 of the rings are planar. Okay, the drug approaches DNA via the major groove, which is another difference from the dactinomycin. Dactinomycin approaches through the minor groove. This one approaches through the major groove. The charged amino group forms an ionic bond. This group, as we said, most of these agents either have charged ionized group or other groups that are capable of forming hydrogen bonding. So this one, the positively charged amino group, will form an ionic bond with the negatively charged phosphate groups of the DNA. Okay, this will lead to a stable complex that will stabilize the complex formed between DNA and topoisomerase. Okay, so it will form a complex DNA topoisomerase doxorubicin complex. Okay, and that will stop the effect of, or the function of, topoisomerase. In the doxorubicin case, it will be topoisomerase enzyme number 2. Okay, and this is the suggested mechanism of action of this drug. And as students at the College of Pharmacy, I think that you like the mechanism of action. Anyway, another mechanism of action for this, that the hydroxyquinone moiety can chelate or chelate, whatever you say, that's fine. Chelate, again, chelate iron to form doxorubicin DNA iron complex. Okay, and that complex will lead to the formation of free radicals, and those free radicals will lead to the strand breakage, DNA strand breakage. Okay, but it is not as important as the complex that form and stop the topoisomerase, but this mechanism is implicated in the cardiotoxicity of the doxorubicin. Okay, also the intercalated doxorubicin inhibit helicases, not only stop topoisomerases, but it also inhibit the action of helicase enzymes. All right, it's easy, just like that. Okay, as we said, doxorubicin is a natural product. It's part of the anthracyclines. Other examples of the anthracyclines are the daunorubicin, epirubicin, and idarubicin. As you can see, their structures here are this figure. Daunorubicin and doxorubicin are considered as the first generation anthracyclines, while epirubicin and idarubicin are the second generation anthracyclines. All right, and also there are some third generation anthracyclines, but they are still under study. As you can see from the figure, idarubicin lacks the methoxy group at R number 1 here. Idarubicin doesn't have the methoxy group. It has hydrogen. Okay, that's why it is more polar and has prolonged half-life. All right, the third generation anthracycline is being studied, as we said. Okay, in the third generation, the aminium ion is replaced with an azido group or a triazole group. This amino is replaced with an azido group and triple bond and, or it's replaced with triazole ring. Such compounds do not appear to be susceptible to the efflux mechanism. Okay, that's one of the advantages of the third generation. It's not susceptible to efflux mechanism, which leads to drug resistance. Okay, and also the third generation shows decreased general toxicity. Okay, which is another example. All right, as I said, if you have a question, ask now. Don't leave it until the exams, okay? Other examples on the intercalating agents, which is different from the anthracyclines, are the bleomycins, okay, which are large water-soluble glycoproteins. Okay, bleomycins are some of the few anti-cancer drugs not to cause bone marrow depression. As you know, most of the anti-cancer, most of the chemotherapy cause bone marrow depression, but bleomycins are some of the few anti-cancer drugs not to cause bone marrow depression. Okay, the bithiazole ring system, as we learned earlier, the dactynomycin has tricyclic system. The doxorubicin and the anthracyclines have tetracyclic system. This one has a bithiazole ring. This one, this group. Okay, this one intercalates with the DNA. Okay, and the nitrogen atoms, and the primary amine, pyrimidine rings, and the imidazole ring, those chelate or chelate ferrous ion. Okay, these nitrogen atoms, okay, these chelate or chelate ferrous ion, okay, that interact with oxygen and oxidize to ferric ion, okay, leading to the generation of highly reactive oxygen species like superoxide or hydroxyl radicals, okay, that results in DNA strand being cut. Okay, as we said, it's just like one of the mechanisms of the doxorubicin leads to DNA strand cut, and we are gonna learn about other examples of chain cutters. Okay, so bleomycin is an example of intercalating agents with chain cutting ability. Okay, what chain? It's the DNA chain, the DNA double strand. Okay, it's either cause single strand cut or sometimes double strand cut. Okay, those superoxide or hydroxyl radicals, how that happen? These radicals abstract hydrogen atoms from DNA, okay, and the result is that the DNA being cut. All right. Also, bleomycin prevent the enzyme DNA ligase from repairing the damage. Okay, so it's not only damage the DNA, but it also prevent the enzyme that repair the damage. That's how bleomycin exerts its action. Okay, it mostly acts or exert the cut happen between purine and pyrimidine nucleotides. Okay, all right. Other examples on the intercalating agents are the mitoxantrone, which is a simplified synthetic analog of the anthracycline. Okay, we all saw the anthracycline. You remember them? You remember their structure? Okay. As you can see from the structure here, this is a simplified structure. Okay, so when we ask what drug development strategy was implied here, it is simplification. All right. The tricyclic ring, the tetracyclic ring system in the anthracycline was, like they call it, pruned or cut into just tricyclic ring system, because as you know, we only need the tricyclic ring system for intercalation. Okay, so they removed the fourth one. All right. Also, this one it is easier to synthesize because, as you can see, it is symmetrical molecule. This part, or part number 1, is the same as part number 2. Okay, so it makes it easier to synthesize. All right, it has 2 identical substituent chains, make the molecule symmetrical, as you can C. It also lacks the sugar ring because the sugar ring was implicated or responsible for the cardiotoxicity that is associated with the anthracyclines. All right, but this still keeps the amino substituents, these, which are the same as the amino substituents available at with the anthracycline. But the anthracyclines, like the daunorubicin, the doxorubicin, those are natural products. This one is a synthetic analog. All right. As you can see from the figure, we have a pharmacophore. The pharmacophore involves phenol groups and the amino groups and the carbonyl groups. You see the one in blue here, the phenol, carbonyl, and the amino group. And because the molecule is symmetrical, it has two pharmacophores. They also found that the amino group linking the side chain to the tricyclic system was also important for activity. This amino group, this one here, this amino group is important for activity. All right, but it's okay that we have two substituent chains, two identical substituent chains, but they found that one of them is fine for activity. So if we delete this part of the molecule will still be active. Okay. All right. Then, as we said, we are talking about mitoxantron or mitoxantron or whatever you want to call it. It intercalates DNA preferentially at guanine cytosine base pair. Okay. And the side chain lie in the minor group. All right. So the intercalating tricyclic system, it will slip itself in the major group, but the side chain will lie in the minor group, and the nitrogen would be ionized and form ionic bonding with the oxygen of the DNA. All right, it is thought to interact with topoisomerase 2 enzyme after the intercalation, just like doxorubicin. Okay. Inhibition of microtubule assembly and inhibition of protein kinase C. These are other mechanisms of action for mitoxantron that has or have been proposed. Okay. Other example on the intercalating agents, which is the last example that we are going to talk about in this part of lecture 1, is the emsacrine, which contain an acridine tricyclic system, as you can see it in the figure here. Okay, and acridine also stabilizes the topoisomerase complexes. Of course, it stabilizes the complex after it intercalates and bound to the DNA. So it intercalate, bound to the DNA, then stabilize the topoisomerase complex, which means it forms DNA topoisomerase drug complex. All right, this is the end of part 1 from lecture 1, and we are going to keep doing that. Each and every lecture, we're going to divide it into different parts. Okay.